Imagine the weight of the Empire State Building resting on every square inch of your body. Not for a moment, permanently. That's roughly the pressure 5 miles down in the ocean. A solid steel sphere sent to that depth, without reinforcement, would crumple like an empty soda can in your fist. And yet, in that exact spot, a small translucent fish is swimming past, completely unbothered. How does soft, fragile flesh survive where heavy machinery fails? Start with the basic problem. Pressure at the surface of the ocean is about 1 atmosphere, the same pressure your body is built for and barely notices. Every 10 meters you descend adds roughly 1 more atmosphere on top of that. By the time you reach the deepest trenches, nearly 7 miles down, the pressure is over 1,000 times what you're feeling right now, reading this. Submarines built to survive that depth need hulls of titanium, sometimes 8 inches thick, engineered by teams of naval architects over years. The fish down there needed none of that. It figured out a completely different solution, and it didn't need a single rivet. The real danger of extreme pressure isn't crushing in the way you'd picture it, like a hand closing into a fist. Water is nearly incompressible, and so is the water inside every living cell. The actual threat is smaller and stranger. Pressure attacks proteins directly, forcing water molecules into the tiny gaps inside a protein's folded structure, prying it open, flattening the precise 3-dimensional shape that makes it work at all. A protein is only useful in one specific shape. Pressure doesn't destroy it, it just gently, relentlessly, unfolds it into something useless. So how do you keep a protein folded correctly when the entire ocean is trying to pry it apart? This is where a researcher named Paul Yancey spent much of his career. Yancey, a biologist at Whitman College, has spent decades studying tiny molecules called piezolites, chemical compounds that deep-sea organisms pack into their cells specifically to counteract pressure. The most important one is called TMAO, short for trimethylamine N oxide. TMAO works almost like a molecular splint. It doesn't stop water from being forced toward a protein's folds. Instead, it makes that water less willing to interact with the protein in the first place, stabilizing the folded shape against the crushing tendency of the depths. Here's the detail that should genuinely surprise you. Yancey and his colleagues found that TMAO concentration in fish muscle doesn't stay constant. It rises in a strikingly linear way as depth increases, more TMAO at greater depth, almost like the ocean itself is dictating a dosage. In a 2014 study, Yancey's team used this relationship to predict something bold, a theoretical depth limit for fish altogether, somewhere around 8,200 meters. Beyond that point, they calculated, a fish would need so much TMAO packed into its cells that its body fluids would become saltier, in effect, than the seawater around it, pulling water out of its own tissues by simple osmosis. The ocean wouldn't crush the fish, its own chemistry would. That's not a hypothesis that stayed theoretical for long. In 2017, a researcher named Mackenzie Geringer, working with a team studying the Mariana Trench, formally described a new species, now known as the Mariana snailfish, found living at depths approaching 8,000 meters, right at the edge of Yancey's predicted limit. No fish has ever been documented living deeper. Not because deeper water is somehow more hostile, in a way shallower trench water isn't. Because the chemistry runs out first. There's a hard biochemical ceiling on how deep a backboned animal can go, and it was predicted with a molecule before it was confirmed with a net. That takes care of the proteins. But proteins aren't the only thing pressure attacks. Every cell in your body, and every cell in that snailfish, is wrapped in a membrane made of fat molecules called lipids. Under enough pressure, these lipids do something you might not expect. They don't get squeezed into a smaller space so much as they stiffen, packing together into a rigid gel-like state, the molecular equivalent of butter left in a cold refrigerator. A rigid membrane is a dead membrane. Nutrients can't pass through properly. Nerve signals can't fire. Cells lose the flexibility that keeps them alive. Deep-sea biologist George Somero spent much of his career explaining how life gets around this. Somero's research showed that organisms living in extreme cold or extreme pressure adjust the actual chemical structure of their membrane fats, swapping in more unsaturated fatty acids, the kind with a kink or bend built into their molecular backbone from a double bond. That kink acts like a doorstop. It physically prevents the lipid molecules from packing together too tightly, keeping the membrane fluid and functional, even while the surrounding water tries to squeeze everything solid. Samaro called this general strategy homeoviscous adaptation, keeping the same functional fluidity across wildly different physical conditions, whether that's Antarctic cold or Mariana Trench pressure. So proteins are stabilized by TMAO. Membranes are kept loose by kinked fats. Between the two, a deep-sea fish has effectively built its own pressure suit from the inside at the molecular level, with no metal involved at all. This resilience comes with a brutal catch, though. A body, chemically tuned for 1,000 atmospheres of pressure, is a body that cannot function without it. Bring one of these fish to the surface, and it isn't relief. It's catastrophe. Without the crushing water pressure it evolved around, gases trapped in its tissues expand violently, and depending on the species, eyes can bulge, stomachs can avert out through the mouth, and swim bladders, in species that still have them, can rupture outright. The Mariana snailfish and its deep relatives don't get lucky by having tough, armored bodies built for both worlds. They're built for exactly one world, and it's the crushing one. Take away the pressure, and the same chemistry that keeps them alive down there starts working against them. There's a quieter cost, too. Building and maintaining this specialized chemistry isn't free. Down in the Hadal zone, food is scarce, sunlight has never existed, and metabolic energy is precious. So these organisms tend to run slow. Slow growth, slow reproduction, slow movement. Survival at extreme depth doesn't just mean tolerating pressure, it means accepting a permanently low-energy existence in exchange for it. Resilience and abundance, it turns out, rarely arrive in the same package. Which raises an obvious question: why would anyone care about a handful of pressure-tolerant fish molecules outside of a marine biology department? Because the same chemical trick nature evolved to stop a protein from unfolding under crushing pressure looks a lot like the trick medicine has been searching for to stop proteins from unfolding under a completely different kind of stress. Diseases like Alzheimer's are, at their core, protein folding problems, misshapen proteins clumping together where they shouldn't. Researchers studying osmolytes like TMAO have been investigating whether these same molecular stabilizers, the ones the ocean invented to keep a fish's proteins intact at crushing depth, might help stabilize proteins prone to misfolding in the human body. It's early science, not a cure sitting on a shelf. But the direction is genuinely striking. A molecule, perfected in the permanent dark of the Mariana Trench, might end up relevant to a disease that has nothing to do with water pressure at all. Modern Hadal exploration keeps adding to this picture. Autonomous landers and submersibles, increasingly capable of repeated unmanned dives to the deepest trenches, are recovering tissue samples and previously undocumented species from places humans have visited only a handful of times in history. Each new organism is, in a sense, another line of biochemical code nature has already tested and refined over millions of years, sitting in the dark, waiting to be read. So the next time someone describes the deep ocean as an empty, hostile void, you might reconsider that framing entirely. It's not empty. It's arguably one of the most sophisticated engineering laboratories on the planet, and it never used a single blueprint, a single weld, or a single sheet of titanium to do it. Imagine the weight of the Empire State Building pressing down on every square inch of your body, permanently, and somewhere beneath that exact weight, something small and translucent isn't just surviving, it's thriving, using chemistry your own cells might one day borrow from.